Hello there, I'm Joe Mandell. I'm a junior studying computer science here at the University of Minnesota. This presentation is called Modeling Methylation, and it's about building Python simulations of biological processes. This work was done in collaboration with Professor Anna Kraut, Professor Jasmine Fu, and the Math Bio group here at the University of Minnesota. I'd like to thank them for mentoring me through this and for allowing me to be part of this really interesting research. So this presentation is about modeling methylation. But to understand that, first we're going to examine what methylation is. Then we're going to look at how Python simulations can help us understand it. Finally, we'll see what relevance these findings have for other researchers. Methylation is essentially a cellular process where small molecules called methyl groups attach to a strand of DNA and silence it. It's like a light switch that can turn off portions of DNA. If you turn off the genes that suppress tumors, for example, you might elevate the risk of cancer. And because of this, understanding methylation will help us understand cancer and aging a lot better. We do have current models of methylation, such as in these papers right here, but the problem is that they're fairly complicated. They're difficult to work with mathematically. So the researchers here at the U are trying to find a simplified mathematical model that will be easier to work with. Where my research comes into that is that I'm creating a Python simulation that they will use to test their mathematical models. It uses something called the Gillespie process, which is an exact simulation of the random biochemical processes behind methylation. And the simulation makes graphs like the one on the right here. I want you to focus on the green and brown lines. These represent the prediction error of those simplified models compared to the actual Gillespie process. Essentially, we can use graphs like this to see whether researchers' simplified models are fitting with the more complex models that we know to be true. And if those error bars are low enough, or those error measurements are low enough, we can conclude that their simpler model fits well, and it's a valid simplification of the existing models. But in order for this simulation to be helpful, it needs to be both accurate to reality and fast enough to run on a researcher's laptop. So the methodology for this was essentially figuring out how to make a simulation that is both accurate and fast. To make it accurate, we just needed to implement the Gillespie process properly. And just an example of how this might look is this graph here on the right, which shows the amount of methylated and unmethylated DNA base pairs. This graph can look a little complicated, but the important thing to notice is that the numbers are never 50-50. For most of the graph, this system is almost completely methylated, right? The red line is much higher than the blue line. And for a brief portion in the middle, the graph is almost entirely unmethylated, right? The blue line is much higher. So this is that bistable behavior that we were talking about. It's, again, like a light switch. It's either on or off, right? Mostly methylated or mostly unmethylated. This matches pretty well with the biological reality, so this is kind of an assurance that our simulation is behaving as expected. I want you to look at this graph again. It looks a little complicated, right? Well, tough luck, we have to make 1 million of those graphs to properly test our theory. That's going to be a little difficult. And in order for that to work, we will need to make this process very, very fast. So this was my main focus for this project, was optimizing the code to make it very fast. The first step was to make it as simple as possible. Then we rewrote the code using Numba, which is a performance-enhancing package for Python that makes basic mathematical operations much, much faster. Then we adapted it to run in parallel. This meant that instead of one simulation running after the other, the computer could run multiple simulations at the same time. This lets us use the computer hardware more efficiently and run, again, much, much faster. This code snippet here shows how we parallelize code in Python using Numba, but let's skip past the Python code and just get right to the results, the interesting part. These techniques made our code 150 times faster, which is huge. This meant that instead of simulations taking almost an hour to run, they were done in less than a minute, making things much easier for the researchers. So, did this simulation work? Did it produce results? That's a resounding yes, it did. Our simulation accurately modeled the detailed system, and it was fast enough that researchers will be able to use it to test their theories within minutes. This means that the biomath lab will have a useful tool to check their theories and work towards a better understanding of methylation patterns. And in fact, we've already used this simulation to study a simplified model for certain parameters, so we already know that it works. There is still work to be done, however. While we found an accurate simplified model for a certain arrangement of parameters, there are many other arrangements of parameters that we haven't explored yet, so we could experiment more with them. We could also try to write some kind of code that would automatically find bistable parameter regimes, like the one that produced that graph with the red and blue lines earlier. We could automatically find parameters that lead to that. The final thing we could do is check against detailed empirical data, like biological observations of methylation, to see directly how our predictions line up with those. So takeaways, what should you remember when you close this video? Python simulations are a great way to start prototyping theories, but you might have issues with them if you need code to be very fast or very accurate. In a situation like this, I would encourage you to consider using performance-enhancing libraries like Numba or NumPy. These libraries are open source, the documentation is great and available online, and they can really speed up your code. So if you're a researcher, if you use Python for hobbies, for research, for work, I highly encourage you to look into performance-enhancing libraries like Numba and just see if it works in your case. In our case, it did, and it made things substantially better for our team. I'd like to thank the U for sponsoring this research with the URS Undergraduate Research Scholarship. And I'd also, again, like to thank Professor Anna Kraut for mentoring me through this, and Professor Jasmine Fu for allowing me to join this research group for the duration of this project. It's been an incredible experience. I'm very thankful to both of them and to everyone else involved for allowing me to be a part of this. Finally, I would like to thank you for watching this video. I hope you found it informative, and I hope you have a great rest of your day.